Today, no history. Today, only biology teachers. Today, we'll start well quickly. Already, we saw that maximum the translation. Everything we completed, we have completed. Only last few lines. Then we'll go to the other concepts, whatever has been told by the group of teachers. That topics only today we are going to discuss. So this translation done quickly. We can recap like we have studied various activation of amino acid, and then all this basic step, then initiation, elongation, termination. What is the role of ribosomes? What is the role of this tRNA? All these things we elaborately we have seen. Now next we are going to see the continuation, last few topic. This is sure that children have understood the topic in a well better manner. So that's why. So how this at the 5' end, what are the things they have to keep in mind is what is the anticodon loop, where the amino acid acceptor end is there, at which end of the mRNA the translation is initiated. So these things they have to keep in their mind very clearly. So how this always, that first one is AUG. Students, even if we say 10 times also, students will always have, at least some students will have the doubt where the AUG will be there. In the mRNA, if AUG is there, what should be the anticodon? What should be the anticodon sequence? That student must be having clarity on that, that we need to make sure teachers. If we are sure with that, that's all. Otherwise, students will be more clear with all the other things. So how you can see that A, P and E sites, how it is joining one by one. There are different tRNA for different amino acid. So how it is moving, how the translocation is happening of the ribosome in respect to mRNA, how the translocation is happening. That also students must be very clear on that. So once it is clear, that how the peptide bond formed amino acid, this already they know in the 11th standard itself, they have studied about it. So they know the basic idea about the polypeptide chain, how the chain is formed, C and N and all. They will be able to definitely say a little. There won't be any confusion in that. So this is for us. So can anyone say, share what is the enzyme used? So it's peptidyl transferase. The enzyme used for peptide formation is peptidyl transferase. It is also in the text. It is important in the supplementary reader also. The topic is there. So
I was
peptidyl
having a
transferase.
doubt, like peptidyl carboxylase something. I was having a doubt.
That
Peptidyl
carboxylase is the place
transferase.
where the initiation, that first amino acid will bind, ma'am.
Okay, okay.
Yes. Thank you.
Yes.
Thank you, teachers. So polysome. So here, students, we need to give a clarity that one mRNA by doing translation will produce a very less quantity. Only one single protein molecule will be formed. That won't be enough to express the phenotypic character of that particular individual. So at simultaneously, many ribosomes will be bound to that mRNA. The many ribosomes on the mRNA, we call it as polysome. That is a very common term. We have been using that. Because of that, what will happen? More and more at the same time, simultaneously, more amount of these proteins will be formed. The polypeptide chains of different protein will be formed at the same time. This concept we call it as translational amplification. Translational amplification. Students will have this doubt always, and also they mislead, they misunderstand. One mRNA that converts into protein, that's all over. That protein is just a single molecule of protein, that won't be enough. So we need to say that at the meantime, even some smart student will ask the question in the class also. So that is because of translational amplification. At the same time, in a many mRNA sequence, that ribosomes will be bound, and same time more amount of polypeptide will be produced. This concept, translational amplification, this is not in textbook. So the smart students, those who are asking, we may have to give that. Next one is teachers. This we can definitely tell to students. Why? Because when they are taking antibiotic, they don't know the idea of how that antibiotic actually kills the bacteria. They simply think that the bacteria, when it's taken, it goes and kills the bacteria. In what way it kills or interfere with the metabolism of the bacteria, we need to little highlight to the children. Of course, we don't have time to explain entire thing. We can give an idea and we can ask the children to even do project in that. In 12th standard they are having project, or even in 11th standard also you can add it in the, when we are teaching about all these antibiotic concepts, we may explain them in biomolecules, we may explain them. So how this actually interfering the streptomycin antibiotic, tetracycline, neomycin, chloramphenicol or erythromycin, all these things are going to interfere with their translation process. Say for example, if streptomycin is given against the streptococcus bacteria, the initiation of translation itself will be stopped. It will read the misreading wrong, in the place of a methionine something else will be there, so it will not be able to read properly. Thus the bacteria will not be able to survive when streptomycin is given. So in this way student must understand, not simply like one antibiotic is given it kills that bacteria. That should not be a very superficial idea with the children. It inhibits or interfere with a specific function of the bacteria. Because of that, bacteria is not able to survive. Same way the tetracycline that interfere with the amino acid tRNA that is failed to bind with this ribosomes. So this we can say the neomycin. So the neomycin, same way the tRNA mRNA interaction is prevented. So in this way we can give an idea how this antibiotic is actually working, how they are actually killing the bacteria. We can give an idea. This will be very important for the student to wide thinking in a very elaborate manner in biology. So this video already we have seen yesterday, so we are not going to see it again. So we'll go to the next directly. Now we are going to another topic which is asked by the teachers only. Two topics we are going to see today. So gene, define a gene. If someone asks, we know very well to define a gene. We say that it is a segment of DNA, it's a cistron, it's an expression sequence, it is going to code for something, it has some work in the phenotypic expression like that, so and so we can say lot of definitions we can say. But now modern concept of gene definition is completely different. Students, it is there in the book also a little, and more we need to even put into them that a segment of DNA that reads as occupied in a specific locus and has a unique sequence of nucleotide base pair. The DNA has a very unique sequence of base pair, or it carries the information, coded information to code polypeptide chain. The gene may be a continuous one or split. It can be a split or continuous one. It's able to replicate itself and also able to produce the phenotypic expression. These are all the modern definitions. Kindly please, either if you want, you can take the screenshot also. Anyway, I will be sharing all this PowerPoint with you, and also recorded video also will be shared to you. No need to worry. But still, if you want, you can take a screenshot now. So now we are going to touch a small topic. Definitely, I request you all the teachers to touch this topic to the children. It will definitely increase their scientific thinking, elaborate their idea of biology. They must know what are the types of genes are there. When the students we are going, they are generally saying two types, the split gene only they will say. They say introns and exon, they will say, and with that, that's all over. That is the maximum knowledge they have. But we need to give more knowledge about the different types of genes. Though we are not going to explain everything, at least a little we can say, for example, constitutive genes, non-constitutive genes, split genes, overlapping genes, single copy gene, repeated gene, multi-gene, pseudo-gene, and transposons. First, before seeing the above two, I will start with the split gene. As split gene is something, like exons and introns, what we have, that is a split gene. Overlapping gene can be asked in the exam how if it is 12 proteins are expressed, but there are only 10 genes are available. In the organism, there are only 10 gene, or in the chromosome, there are only 10 gene, but the chromosome contribute for 12 different type of proteins. From that, we can easily say the gene we are there is called as overlapping gene. Otherwise, how come a 10 gene can contribute for 12 different type of proteins? So overlapping gene, we can give an example. Single copy gene. The gene will be only one copy of that will be there in the around 60 to 70% of the chromosome. Those genes we call it as a single copy gene. And repeated gene, same gene will be repeated many times in throughout the chromosome, such we call it as repeated gene. Multi-gene, a group of gene will work together to express some phenotype. One gene cannot do it all alone. A group of gene which will be more or less similar will be expressing a character. Pseudogene, which is not actually going to express anything. Transposons, you can say very well what are they commonly called as, teachers? Transposons.
Mobile genetic elements, sir.
Very nice, ma'am. Thank you, Saraswati, ma'am.
Yes.
Yes. Can anyone say the application of this in that, the
The
nematode?
RNA interference technology, this transposons is used in order to create antisense.
Very nice, ma'am. Exactly, ma'am. Exactly. Yes. The transposon you would have seen there.
Why, sir? Because this particular concept, I was tired of teaching to the students every time. They used to get confusion in this nematode resistant RNA interference technology, because those are all we have taught during the online classes. No, again, the students will be getting doubts only in this particular topic, especially this biotechnology chapter.
Yes, definitely. You are right, ma'am. But what happens, even if in the offline students are teaching also, if we do also, the problem is comes because of they have no idea about gene. They are having a blind opinion about the gene. Gene means a part of a DNA that is going to express. When we say the transposon mobile genetic elements, they are not able to comprehend that what exactly it would be. So if we give an idea of different type of genes available in the in an organism, now there will be somewhat more clarity. They will try to understand what transposon. That's why we have to teach it during the molecular biology itself. That will not interfere with biotechnology. Biotechnology, they will be very clear if we are teaching these transposons in this molecular biology itself. Even I had the same experience in the beginning. So after that, now it is mostly the students are clear about it. So different type of streams you can see. So what are constitutive genes and non-constitutive genes? Means they are nothing but the constitutive genes are called as the expressing, what is going to express in our body, whatever the phenotypically it is going to express, those genes are called as constitutive genes, which is definitely mandatory for our body normal functioning. So we call it as constitutive genes. Whereas the non-constitutive genes also called as special genes, luxurious genes, different names are given. So they will be only expressing if there is a need, otherwise they won't express. If there is a only need, it will be expressing. If there is no need, it won't express. Such genes are called as non-constitutive genes, whereas the constitutive genes will be all the time expressing. So now small question for you. So can anyone say which genes are called as housekeeping genes? Yes, teachers, you can try. Just put your, it's definitely not indirectly the reader, but we would have known based on the previous slide. You can try to. Anybody would like to try? Why Jansi ma'am is very silent today? What happened? Always ma'am will be giving answer first. Ma'am, are you there?
Highly conserved part of genome.
Okay, ma'am. No issues. Nothing but the constitutive genes only is called as housekeeping genes. That is very important for the body functioning. They are very essential. So we call this constitutive genes only as the housekeeping genes. Now another question then. The operator we are studying now, we are going to see now, that comes under which type of gene? You may try to say that. the operator molecule, which we are saying that we are going to see different operators. That one operator we have in a 12th standard. That comes under, what do you think, which gene category would it come under? It expresses only when there is a need, otherwise it doesn't express. So now you can say.
Non-constitutive gene.
Yes, very nice, ma'am. Yes, it's a non-constitutive genes or luxurious gene or special genes. All the names are same only. So this gene operator comes under non-consecutive. So this way, if the students are knowing different type of genes, definitely their idea about the NCERT book, the whatever is given in that, that will be much easier for them. So this many gene names, if they are, of course, it will be little, it will take some or 10 minutes extra, 10 minutes extra for us to explain all these things to the children. But once they learn it, here also they will be using it in the operator also. And same way in the biotechnology also, whenever the gene concepts are there, they will be able to use it. Same way, this split genes also, once we explain, that will be useful for them in the various chapters. It will be very much useful, teachers. You can just try it out. Maybe this year you can try to use it. So regulation of gene expression. What is regulation? Student will be always having doubt why it has to be regulated. So we need to very give a clear idea. The formation of a polypeptide or the expression of characters will only happen if there is a need. Otherwise, they don't need to express. Say students will immediately relate all this thing with the Mendel's law of inheritance. What if the gene is not expressed? Then what will happen? What will happen to this color of the seed, color of the flower? So students will relate to that. So we need to bring the student out of that mentality, and we need to give a best example within ourselves. Say for example, when we were a small child, during digestion, we need one enzyme, but when we grow up, that enzyme is no more required. Can anyone say which enzyme?
Renin.
Yes,
Renin.
ma'am. Very nice. Yes, yes. So it is not required. So that idea, if we give it to children, they will have some idea. Yes, there whenever there is a need, that enzyme is produced. Otherwise, it is not produced. So eukaryote and prokaryote, we need to give a clear-cut differentiation to the children. In eukaryote, the expression can be regulated at 4 different levels, that including transcription level, processing level of splicing or capping, or the transport level from mRNA from nucleus to cytoplasm at the time, or translation level at the time the translation will be stopped. In any 4 stages, the regulation can happen. Once the transcription has happened, later if it is decided there is no need of that particular protein, then processing will not happen. Those expressing sequence will be converted as a non-expressing sequence. In hnRNA itself, it will be removed, spliced out. It will be removed out. Or after splicing only, we come to know that body thinks that there is no need of that hormone or enzyme, that particular protein. Then the transport, during transportation, that particular sequence will not be transported. Or during translation level, maybe that formation of polypeptide will be interfere or stopped. These are 4 different levels are there in the expression of gene in the eukaryote. Here student must understand, eukaryote is a very complex organism. So they have complex chromosome structures. Whereas prokaryotes do not afford to have such a kind of elaborate mechanism. Why? Because their body structure itself very primitive. The number of gene in their body is very few. So in that case, they not afford to have this much complex mechanism. They have a very simple mechanism, like in our house we have switch on, switch off. When there is a need for something, the mechanism is switched on. When there is a no need for that, then it is switched off. We need to introduce the concept. Can you say, teachers, which concept we are going to study? We are going to see now, discuss now.
Regulation of gene expression.
Yes,
The
what is
lack
that
of-
example is?
Lack of lack of
Very nice. Lac operon is the thing we are going to study today. Lac operon, it's all applicable only to eukaryotic or the prokaryotic organism. Eukaryotic organism, students must be very clear. It is not going to affect that. It is not in the eukaryote, only in the prokaryote. So first of all, as I always say, credit must go to the scientists who actually discovered that. So it is Jacob and Monod, these two scientists, very important discovery they have made in the bacteria about this. Yes, ma'am, Saraswati, ma'am.
Sorry, sir. Sorry, sir.
It's okay, ma'am, no issues. So what are all the components? Whenever we are saying to the student must keep in their minds very clearly what are all the components are there in this operon. Any operon, it's not necessarily lac operon. Lac is one example we are going to explain to the children. But whatever, whenever we call it as an operon, what are all the things should be there? In any operon, there must be a structural gene, there must be a operator gene, a promoter gene, a regulator gene, a repressor, and definitely an inducer. This all the things constitute a operon concept. Whether it is a lactose or it is a tryptophan or whatever that may be, first of all, these things are very, very must. These are all the structure present in the operator concept. The types of operator also we need to say a little, because if we say here the negative regulation, positive regulation, students will have a clarity. So what are the two? If you see inducible and repressible, inducible and repressible. Inducible is what we are going to see now. Inducible, in which what will happen? If inducer is there, that will work out. If the inducer is not there, the mechanism will be switched off. In repressible, what will happen? Generally, the mechanism will be happening. When repressor is given, it will be switched off. When repressor is not there, it will be switched on. Just keep in mind, students put it in a better way. In inducible one, when an inducer is there, the mechanism will be there. It will be switched on. An inducer is not there, it will be switched off. When repressible, if a repressor is there, it will be switched off. If repressor is not there, it will be switched on. So negative and positive mechanism, in this way we can explain to the children. So lac operon, which we are going to study now, it's an example of an inducible operator system. So this diagram and all very clear. We know very well how that operator is there. Operator is next just adjacent to the promoter. Then we have the structural genes are there. Z, Y, and A are the structural genes. And here also one promoter is there. Here I gene is there. This promoter is going to promote the transcription of this particular I gene alone. The I gene, students always get confused because inducer is the word we are using. Students will get confused. It is as an inducer. So again and again, we need to put into their mind, I doesn't stand for inducing. I stands for inhibiting. So inhibiting molecule called as repressor molecule. We need to put it into the mind of children teacher. This, whatever happens, this inducer molecule is going to, the repressor will be produced. The repressor molecule will be definitely produced. So now operator will determine whether the structural gene is going to function or it is not going to function. That will be decided by this particular operator gene only. If the repressor binds to the operator region, then transcription of structural gene will not happen. If repressor does not bind, if the operator is free, then the transcription of this 3 structural gene is going to happen. Who will decide the blocking of operator by the repressor? Who determines means it is nothing but it is the inducer. Inducer here is lactose. The lactose which we are going to give is the inducer. If the inducer is not there, the repressor is free to bind to the operator region. If inducer is available, it will going to lock the repressor and inactivate the repressor, so that operator molecule is free, so that structural gene will be expressed. So again, this student, this inducer, what happens in the absence of inducer, what happens in the presence of inducer, student must have that clarity. Now, can you say the teachers, what is the function of Z, Y, and A gene very quickly? We shall go to the next one then. Yes, teachers.
Beta galactosidase is that production of enzyme.
Yes, ma'am.
Production of enzyme permeases, transacylases, and beta galactosidase.
Very nice, ma'am. Yes. Z gene is going to code for beta galactosidase enzyme. The job of enzyme is to, here student must understand, the galactosidase will break down this lactose, hydrolyze the lactose into glucose and galactose. Now we need to again say to the children, if lactose is not available, what is the need of beta galactosidase that we need to again and again put into the mind of children. Then Y gene that codes for the permease, which is going to enhance the permeability of lactose into the cell. And A gene is going to code for the transacetylase. This is very important for the smooth functioning of this the metabolism of lactose. All the three genes are very important for lactose metabolism. Now, even in the absence of one gene also, the lactose metabolism will not happen. All the three genes are mandatory. Now we'll see about it. It's very quick only. Already it's a very established, very easy concept. So operator, the operator gene is actually the before all this structural gene. It is located adjacent to the structural gene. The operator gene located. Student must definitely write that sentence. It carries the mark. So it located adjacent to the first structural gene, that is that gene, and it always remains switched off only in the presence of, it always remains switched off in the presence of a repressor. When a repressor is there, it is always switched off. You all the time. When the repressor is not there, because of the inducer, then only it is switched on. So student must always write that the operator gene is always switched off, because there is no inducer is there all the time. All the time the job is it is switched off only. That's why we call it as again negative mechanism. Next, it is actually the operators. A little more students may write it. It is not a textbook concept. So the length of the how many base pairs are there in the this operator is 27 base pairs are there. So same way promoter. The promoter gene lies left to the operator. They have to definitely write the location. It is located left to the operator. And the main function, how the promoter is functioning. What is the job of promoter? It is the site that is going to be recognized by the RNA polymerase. It is the site that is going to be recognized by the RNA polymerase. The operator also, the promoter also, both are a palindromic sequence. Both the side, when we read, the same type of nucleotides are arranged. So both are the palindromic sequence only. Here itself, we can say what is a palindrome, so that when they are studying biotechnology, they will not have confusion. So it's a palindromic sequence. Then I gene, the regulator gene. This job of I gene, again, as I said, students will again keep in mind as inducer. So I gene is actually an inhibitor or a repressor gene. It is going to code for repressor. All the time it is going to code for repressor. Whether the lactose is given or not given, in the presence or absence, always a regulator gene is going to be expressed. It's all-time expressing gene. It is going to produce a repressor protein for sure. The binding of repressor protein. Yes, ma'am. Namaste, ma'am. Am I audible, ma'am?
Yes, sir. Yes, sir.
Yes, thank you, ma'am.
So now only I came, sir.
It's okay, ma'am. It's okay. So structural gene is there. So this is what are the structural gene, operator gene, regulator gene. If this table we show to children, they won't have any doubt for the exam. In an exam, they will definitely write very well. Very clear differentiation is given in this particular table. What is the job of operator, promoter, regulator, and structural genes? So they need to have that clarity of all the different type of genes. That's it. So we have completed operator. Why can teachers, anyone say why it is called as negative regulation, teachers? Why not it is positive? You can try, we'll also join.
Sir, it is because repressor is inhibiting. By inhibiting something, if a gene is getting regulated, we call it as negative control.
Very nice, ma'am. Exactly. By inhibition, as I already have said before also, all the time the repressor protein is going to be produced, that is going to region, that always the mechanism is going to be switched off only. When an inducer is given only, the mechanism is going to be switched on. So when the inducer is given, it is switched on, otherwise it is switched off. We call it as a negative response. In a positive regulation, what happens? All the time the mechanism will be switched on. When we give a repressor, it will be switched off. Then we can call it as a positive regulation. That we need to, though it is not there in the last one sentence only is there in the reader, that we need to inform to the children. Shall we go to the next topic, teachers? Human Genome Project. Are you ready?
Yes, sir.
Yes, thank you. So it's all teachers requested topics only. Today we'll try to complete human genome as well as the fingerprint. Both will try to complete, teachers. So Human Genome Project, it's a very ambitious project, as we all know that. When we tell to the children, there are half of the topic in that this memorizing. We need to memorize, make the children to memorize only, because that's all facts and figures. What is the biggest gene? What is the highest number of nucleotides? Where the which gene is a very having average nucleotides. So all this data students can only memorize. There is nothing much to explain, but other 50% where expressed sequence tag, what is that sequence annotation, the methodologies, there and all when we study, definitely the student must very clearly understand what is that Human Genome Project, what are the methodologies employed, they must be definitely able to understand. So that DNA, it's a very, as I already said, it's an ambitious project. So we are going to, DNA is packed in 23 chromosomes. They are going to sequence 23 plus 1. Why I say 23 plus 1? Can anyone say, teachers? They are going to sequence 23 plus 1 chromosomes.
Because total autosomes are 2. I'm sorry, sex chromosomes are 2. For male 1 and female another an extra, maybe for that.
Yes, ma'am, exactly. But in the way 23 chromosome, in that 22 autosome and 1 X chromosome plus Y chromosome separately. Out of 22 pairs of autosome, we are going to take only one pair only. One out of one pair we are going to take. Whereas in the X chromosome, Y chromosome separately we
Pair
have
or
to take.
23
Sir,
chromosomes,
excuse me,
sir?
sir.
What, ma'am? Sorry, ma'am.
Is it pair or 23?
23 single chromosome plus 1 Y chromosome.
So actually
In the
22
23 yes, ma'am.
Actually 22 plus XX or 22 plus XO idana sir varum pass la.
Ma'am, actually 22, when we say that what is the number of autosome and sex chromosome, we'll say 22 plus X and X, 22 plus X or Y, like that we say. But
Yes,
when we are sequencing, 22 single autosomes we are going to take, plus X chromosome we are going to take. Without a pair, Y only is there separately. To denote that, they will say like 23 chromosome plus 1. That is actually like a formula they use that while sequencing. That is a common word they use, 23 plus 1. That is what they will take it for sequencing, ma'am.
One more reason is that you have given only as a haploid, only in gametes, like haploid cells only they are given. That's why it is written as
Very
20.
nice. Yes. Thank you, ma'am. Thank you. So it's again an ambitious project. Why we say ambitious project? When children are understanding, in a book it is given an elementary idea, 2 pages, it is over. But when they are doing it, actually it's a mega project. Why? Because simultaneously many scientists had to involve in that. A sophisticated technology, various molecular, whatever the well advanced technologies were there, those technologies were employed in that. And sequencing a human genome took nearly of 13 years. They planned the goal itself for 13 years. So because of this various reason, we call it as the Human Genome Project is a mega project. So it is lot of fund also involved in that. Billions of amounts are involved in that. And another very biggest
Many
challenges.
countries also.
Yes. Yes, ma'am. Yes.
Many countries, around 130 countries were also involved in this.
Yes, ma'am. Yes, yes, exactly. And also this computer bioinformatics, here we can give an idea of career counseling about bioinformatics to the children. The bioinformatics, that is all the sequence cannot be stored in the database of the book. Actually in the book it cannot be stored. So computer, they have taken the help of the computer to store the data. If we store alone the data, how come we take it and study whenever there is a requirement? So for that, retrieving the data from there, whatever the data is stored there in the computer, that has to be taken. For that, separately tools are there, various softwares are made. So all these things will come under bioinformatics. So because of all this reason, we call it as a mega project. So what is the main idea of this Human Genome Project? Means they have to map the human genome, they have to sequence, they have to analyze the function of each. And when it comes to human, I don't know why, when it is an animal, we don't talk about the ethical reason. When there is something against a human, whenever some project involves human being, definitely many people will come up with the ethical this protest. So ethically we need to address that. Second thing, when it involves human, the biopiracy, this kind of the misuse of the data and all can happen. That and all needs to be addressed, that has to be avoided. So for this reason, the ethical reason also has to be addressed. Apart from doing the mapping, sequencing, functional analysis, but when it comes to human, the ethical reason also need to be addressed. That's the main objective behind this Human Genome Project. Here itself, we need to tell the students why that human involving, what are the ethical problem would arise in the future. The data, even when we collect the Aadhaar data, we have a problem. Why Aadhaar data is collected, Aadhaar will be leaked into the public, what will happen? Same way in the Facebook, WhatsApp, and all this thing, we have the concern of the privacy. Same way when it comes to the Human Genome Project, that much data is collected. If the data goes into the hands of the people, those who want to destroy the things and the destruction mentality, so what are the challenges will be faced? How come that that can be avoided or prevented? All these things we need to definitely put into the children. Then only they will have the broad thinking about the ethics behind all this project involving either human or any other animals also. The sequencing is done by, as we all aware, two things: the expressed sequence tags and sequence annotation. The expressed sequence tag is very easy method. In that, nothing they will do. They will take the mRNA. They will directly target the mRNA. Why? Because the mRNA containing mostly the expressed sequence only. Then they will convert those expressed mRNA into the DNA and they will sequence it. They will directly target mRNA, not the DNA. Why? Because DNA is comprised of both exons and introns. But when we go to this mRNA, only the expressing sequence are there in the mRNA. So mRNA will be taken. That mRNA will be again converted into DNA. Only the expressed tags will be there in the DNA. For that, they will be sequencing the alert the sequence. That we call it as expressed sequence tags. Whereas in the sequence annotation, they will not go with such a kind of thing. They will bulk DNA, whether it's a coding, non-coding, a mixture of exon and intron, they will isolate. Then after they express in the bacteria, they will assign the job for different protein features. Little elaborately, I have put into for your consideration, for your easy then. Expressed sequence tags were discovered by Donis Keller and Craig Venter. These two scientists have to be given credit for that. Donis Keller only actually find that RFLP. Can anyone say what is RFLP, teachers? It's a very important prerequisite. RFLP.
Fragment length polymorphism.
Very nice, ma'am. Restriction, fragment, length, polymorphism. Restriction, fragment, length, polymorphism. This idea is very important behind expressed sequence tag. Then they isolated, as I already said, mRNA. That mRNA is converted into DNA. The DNA, based on that, what is that mRNA, whatever the protein is going to be made, that they will sequence. You won't believe this scientist by 1992, this scientists have actually sequenced more than 80,000 genes. These two scientists together, they sequenced more than 8,000 genes by before 1992. And even they have completed the physical mapping for all those things by 1994 itself. So that much best method is there. But on the other hand, sequence annotation also important. Why? Because human beings are the recently evolved organism. Very quite recently we have evolved. In our body, we have expressed sequence also. Non-coding sequence also is there. As we all know that this non-coding sequence is the very important thing that determines what is our evolution history. So if we want to know that, then only we can predict where we are going in the future. If we want to know where we are going in the future, we must know where we have come from. So this sequence annotation also very important in our body, since there are many DNA which are non-coding DNA also is there in our body. In this, they will isolate the bulk DNA. They will make it into fragments. They will code the DNA into the particular vector, maybe bacterial vector or yeast chromosomal vector, and they will find the protein expressed in that particular from that particular gene. Here we need to understand very clearly, if the scientist doesn't know about the protein structure, it is not just a biologist alone involved, even the various chemists also involved in that. Why? Because the detailed understanding 3D structure of a protein is very, very important. If we know the structure of the protein, then only we will be able to identify which protein is expressed by that particular gene. Otherwise, we won't be able to distinguish between different protein, and that entire process will be a huge failure. So knowledge of protein structure is very important whenever we are doing the sequence annotation process. So general methodology, how it happens. A DNA is isolated from various cells of the body, that is converted into fragments. These fragments are put into the various vectors. It can be bacterial artificial chromosome vector or yeast artificial chromosome vector. Then it is amplified. After that, it is sequenced based on the method developed by Frederick Sanger. We are going to see that what method he has developed. And also in the exam, that question may come, that he also has contributed for one more thing. Can any teacher say that? He has contributed for one more thing also, Frederick Sanger.
Sanger and Nicholson. Sanger and Nicholson.
Yes, ma'am. What is that, ma'am?
Determination of amino acid in a protein.
Excellent, ma'am. Yes, Amada, ma'am. Thank you. Yes. So in the protein, the amino acid sequence, they are all in a proper sequence, isn't it? That sequence was also formulated by this person only, Frederick Sanger only. In a protein, what are the sequence of amino acid is there, in what sequence it is there, that also formulated by Mr. Frederick Sanger only. Next, this sequence will be arranged, whether there are any overlapping is there. If overlapping is there, that also will be taken into consideration. Then all this sequence will be computerized. Manually, it will be computerized. Manually, it will be put into the database. There are different websites. You also can go and search in the website when time is there, like NCBI, NLM, like that website is there. NCBI, you can put it in the Google and you can find it in that. There are many databases you will find. Even teachers, you will find the genome map. Very nice genome map. Map is given. If you go and touch on the chromosome, wherever you touch, it will show the instruction about information about that particular gene. In a very nice way, it is given actually in that. When time permits, you can also go and search it in the NCBI website. So next one is this Frederick Sanger, which I have said, how the Sanger actually identified what is that. Otherwise, we cannot find what is that adenine, guanine, cytosine, thymine. How come we know what is which nucleotide sequence is there? How come we know? That is a question students will always carry in their mind. So it is done so by using a fluorescent method. He used a fluorescent dye. Using that, what will happen? He has made whatever the DNA isolated, he will break that nucleotides, will mix with the fluorescent dye. Different fluorescent dye will join with the one particular nucleotide only. Say for example, if it is red color dye is there, means that red color dye will always bind with thymine only. That yellow color dye will always bind with guanine only. The green color with the adenine and the blue color with cytosine. After that, he will be studying in the sequencing, the chromatogram. When he study, he will have the peaks, different peaks. But what color peaks he is obtaining, from that he will come to know what is that particular nucleotide is. If yellow color is like, you know, GGAA is there, means the sequence will be like yellow, GG, yellow, yellow, green, green. If he is obtaining in that way, then it is a GGAA sequence. Like that, he will give the assignment to that particular nucleotide sequence. This knowledge, kindly put it in the children, because they always have this doubt. How come it is possible after the expression in the bacteria? How come it is possible for them to, they simply given in the textbook, it is sequenced based on the method developed by Frederick Sanger. So kindly students must know what method is developed by Sanger. Kindly you also put it into them, so that they will have a clear idea, somewhat idea will be there in the children. Next one is, it's a diagrammatic representation, how actually that is done. So from liver cell, for example, taken the DNA, it is made into fragment, it is expanded into a clone. Then from the clone, using the Frederick Sanger, it is what is the sequence identified. Later, it is computerized. So this is a simple depiction of the process. Though it is looking simple, the process was not so simple. It is a highly time consuming, manpower, machine, all highly consuming process. Then this, how come this biology, the students must understand, this computerized data is over. Okay, in the computer we have stored all the sequence, very nice. Now how come, what is the use of storing in the computer? Are we going to just simply keep it in the computer? Well, how then better we can write it in the book and we can keep it out. What is the use of that? By simply storing in the computer, with that our job is not over. Now say for example, tomorrow a new human has been evolved. We come to know there is a evolution is there happening in the human. From that person, when we take the DNA and what is already stored DNA in the computer, when we run both the things together, we'll come to know where at what point or which gene the evolution has happened, we will come to know. Or we want to know how much similarity is there between human and the chimpanzee. For that also we can retrieve the data very easily by putting that chimpanzee genome and human genome, comparing in that computer. We'll be able to say how much, what are all the genes are 100% similar, what are all the genes are different, that we will be able to know. For this, what is possibility, how it has become possibility, if you ask, it's called retrieving the data. We need to retrieve the data from the computer. That human, we cannot normally, without the knowledge, we cannot normally retrieve the data. It needs a special knowledge of bioinformatics. A person should have, starting with C++, PHP, Java, all this computer based thing also. And also the person should have the knowledge of biology. Both the things combined together, if the person has, the person only will be able to retrieve the data from this computer. So this is called retrieval of the data. So for that, there are many software developed by the different companies. There are many tools are there. By using the tools, we can take the data and we can view under in our laptop or in our computer. We will be able to see that. There are many other facts are there. Actually, when the genome project was completed, it was actually the 50th anniversary of Watson and Crick, after they discovered that structure of DNA, the double helix structure. After they discovered, on the 50th anniversary, that same year only they have completed the project, this human genome project also. Around in the 2000 year 2000, it was roughly the human genome project declared as completed, but it's a rough completion only, not a complete, fully it is not completed. And various journals and all, it was published, and it has created various outrage from different people, segment of people. They felt like the human population is going to become extinct, like different myths were there, that will not enter. Then more than the genes, whatever they cloned, they were all very length in their base pair. There were many base pairs were there in those genes. Then we can say the last chromosome, the chromosome number 1 was completed in May 2006. Only it was completed. So totally 22 autosome, that 23, that is X plus Y separately, that also has been completed. Based on this, they have even sequenced various other organisms, including bacteria, yeast, and various other organisms are also have been sequenced. Now, in the current status, it has been completed. So we can show to the children what is the milestone achieved in this human genome project, whether they have had goal, whether they have achieved according to that or not, or is there is any variation in that we need to tell to the children. This particular table will give a clear indication about whether the goals are accomplished as per the plan or not. So first one is they wanted 2 to 5 centimorgan resolution map for around 600 to 1500 markers. They wanted to do it, but what they have achieved is they have achieved for 3000 markers itself with 1 to 1 centimorgan resolution map. Can anyone say what is 1 centimorgan resolution map, teachers? Or whatever, even what is centimorgan? You can try. Morgan, we can say, what did Morgan do? You all know what Morgan has did. So based on that, can we try to say something?
He tried to show good recombination.
Yes,
Varieties.
ma'am. What did he say? If the genes are
How much distance the gene has been located, that much amount of recombination characters will be there.
very nice, ma'am. Yes, the gene, if they are located very closer, they show more recombination. Am I right, ma'am?
Sir, more farther.
Yes, when they are located together, they show more linkage. And when they are located very farther, they show recombination. The gap between two gene, the distance between two gene, the measurement is used called as centimorgan. Centimorgan is the unit we use it to measure the gap between two sequence gene, the gap between two gene. So this, like in this way, there are various things what he has discovered, whether the goal was there, according to that goal they have achieved or not, they have it's given the clarity. So which year it was completed also they have given an idea. This you can take it and can make use it in the school or class also. So salient features, which is actually again it has to be memorized by the children. There is nothing they have to understand here. Mostly it is like memorizing only. Teachers, whether the methodologies, are you clear, teachers? Do you have the confidence hereafter student will know very well? Your class student? Yes, you do. Yes, thank you. Already you are doing. Some teachers has asked that more clarity. I hope it will be given. Next. So the human genome contain around 3164.7 million nucleotide bases. The total number of gene actually only 30,000. Actually, previously it was estimated. Do you know? Remember, teacher, how many it was estimated? How much it was estimated, teachers? Am I audible?
Yes, sir. Yes, sir. Audible.
Yes, yes. Previously, it was estimated how much? Do you remember?
No, sir.
Around 1 lakh Am I right? More than 1,25,000 it was actually estimated, but was found only 30,000 gene only. It is there in our body. So on average, each gene will have around 3,000 bases, but it is actually it's average only. Some will be much bigger, where some will be very smaller. Best example, dystrophin on X chromosome, which has around 2.4 million bases. This and all children will have to memorize very well. Memorization only is important. If the students are clear here, this if we clear, then fingerprinting they will understand very easily. We need to put it into the mind of children. Each and every individual is similar by 99.9%. We are all similar. Only around 0.1% we are all different. That we need to put it in the children. Then only they will be able to understand the fingerprinting concept. And we don't know what is the function of over 50% of the gene in our body. This and all student need to appreciate the effort made by the scientist to discover everything. And in future, they may become one of the scientists who may discover the function of one of the gene also. Any 1 or 2 gene of this function also, if they discover, it will be a proud moment for all of us. And the chromosome 1 has the most number of genes, where 2,968, whereas the Y gene has only very few genes, 231. This and all a student must be able to understand. And here itself we need to tell the children about the single nucleotide polymorphism or SNPs gene. If the student understand here, the student will be able to little easily understand that the next topic, the DNA fingerprinting teaches. So every individual, the repetition, even if you say that 2 different genes, if we compare one gene, the other gene, there won't be very big different and all. Maybe one nucleotide will change. One nucleotide will change from one person to another person, then we call it as a different gene. Or if you take from human, one human and another human, physiologically, anatomically, we are all same only. Very 0.1 gene is like out of all the books, one book in one page, there is a different nucleotide is there. That because of that, it makes 2 different individuals. Same way, this human genome project very important to understand the disease associated sequence of tracing human history. If we say that recently also the study was published, why these people, those who are in Europe, they were getting more corona compared to the Asian people. In Asia, the impact was much lesser compared to the Europe. How come they come to know that why that Europe has more impact on this? If we see, they have a defective gene, a disease resistance. Naturally, they have poor because of genetics. They have taken it, they traced it very well using the genetic map. So that also we can put it in the students. So students will be able to have more elaborate understanding on this particular topic. Teachers, shall we go to the next topic, fingerprinting?
Yes, sir.
I didn't touch any history. Am I right today? So
Yes,
if you want history, we'll add on that also. Yes, thank you. So DNA fingerprinting, next very important topic for the children. This year we can expect all these 5 more questions. Human Genome Project. Luckily, if the question asked, student will get 5 on 5. If it is DNA fingerprinting, some under this, that experiment. DNA is a genetic material experiment. Then it will be little. We need to prepare the children very well for that. So DNA fingerprinting, most expected question in the board exam all the time. So that also you can see. So what is DNA fingerprinting? It is like how our palm print is very unique to us. Same way our DNA also very unique. But the DNA will be matching with a very closely related people only. Though we are all 99.9%, we are all similar, but no 2 individual will have the same DNA sequence or same type of DNA. That is what is explained in the DNA fingerprinting, and also which is called as DNA profiling. In our CBSE will always ask such a twisted question only to put the children in confusion state. So we need to tell the children it can be even called as DNA profiling. It is developed by Alec Jeffreys, the scientist who developed in 1985. Very recently only it was developed. So we need to put that. Before we start that, children must understand few things. What is mean by repetitive sequence? What do you mean by VNTR? That is microsatellites and the macro satellites and all. What are the satellites are there? What is repetitive sequence? Student must understand. So before we start that itself, we need to put a question to children that when we are all having similar DNA, how come we find who is the, maybe in case of paternity testing, how come we'll know who is the parent? How is it possible? We are all having 99.9% same DNA. When we put this question, it will make the children to think. They will come up with various answers. And we need to explain it is based on some philosophy or principle called as a repetitive sequence. Mostly these are non-coding sequence only. They are repetitive. That how many times that particular sequence repeats, that is what is called as repetitive sequence. This won't be same for two different individuals who are not related. It will be somewhat similar to the individual who are closely related, and it will be mostly or 100% similar. Can you say, teacher, to whom? In which case it will be 100% similar?
Identical twins, sir.
Very nice, sir. Yes, identical twins, homozygous twins, it is definitely similar. So children must understand it is not based on the similarity we are going to test. We are going to test the paternity or in this DNA fingerprinting based on how much we are different from each other in terms of repetition. We are going to find the fingerprinting based on how much differences are there in two individuals in terms of the repeated sequence. You can see on the screen also, a person one has the sequence of 8 times it is repeated. In other, it is repeated 9 times. In some other, it is repeated even 10 times. So different times it is repeated. So we can say that their three individuals are differing in their particular repetition. Otherwise, their nucleotide sequence are going to be same only. So these students must understand. Okay. Now, how it is done in the laboratory, how it is done, the methodology of doing. First job will be done, the scientist will, or the forensic expert, whoever is involving in doing the job, will isolate the DNA from different individuals. They will make it into density gradient centrifugation. This density gradient will separate the DNA according to different layers, which we actually different peaks will be there, that we call it as satellite DNA. Satellite DNA will not be similar. It will be having different number of repetition. On the based of the number of repetition, it can be called as microsatellites or mini satellites, like different names are given. Say for example, microsatellite and all, if you see, only 2 to 6 base pairs will be repeating. Only 2 base pair to 6 base pairs will be keep on repeating. Such a base pairs we can call it as microsatellite. We even called as short tandem repeat, STR. But for our DNA fingerprinting, STR is not useful, that is not required. For our DNA fingerprinting, we need something called as VNTR, variable number tandem repeats. In what way the variable number tandem repeats differ from that macro satellites? If you see microsatellite, it will have more than 100 bases will be repeated. The 100 base pair in each segment, more than 100 bases will be there. That will be repeated for many times. So that's why we call it as it's a mini satellite. It is not a micro satellite. Micro satellites, we cannot use it for DNA fingerprinting. We need to use only mini satellite, that is like variable number tandem repeat only we can use for DNA fingerprinting. Again, we need to put into the mind of students, in the variable number tandem repeat, hundreds of bases will be repeating n number of time. It can be 4 times or it can be 10 times also. The number of repetition can differ, but in each segment at least hundreds of bases will be there. Whereas if you say take microsatellite, it will be just 2 bases, 6 bases, 5 bases will be there. When 5 bases and all is there, we cannot study them properly. We cannot compare with two different individual. So we need mini satellite only, that is VNTR. Now what is happening? Person 1 VNTR, person 2 VNTR. Adenine, many adenines are repeating, many guanine is repeating, whatever that we are going to make it as a segments. We are going to compare with the two different individuals of the repeated sequence. But before, how come we repeat it? How come we visualize? Students will have the doubt. For that only, next we are going to see that this will be repeated with the different methods. The methodologies will be used. Of the 6 different steps will be used. The first step is the DNA will be isolated. It will be isolated from any of the sample. It can be blood stain, or it can be hair, or it can be semen, or it can be saliva, whatever the sample. In case of forensic evidence, blood stain and all is very important, or in the bone or other things. If a person is completely killed and is not able to find his face or anything, then we can take the person's bone, or bone. Mostly we'll go with the bone only. So like that, different samples can be collected. From the DNA has to be isolated. Student must be very understanding here. Here itself we need to tell about some examples of various forensic investigation, or some other thing we can give as an example, so that children will definitely keep it in the mind very clearly. Next, after whatever we have isolated, we need to, same way like how we do in the biotechnology, we need to fragment it using some restriction enzyme. Then we need to run it in the gel electrophoresis. We need to make it run in the gel electrophoresis. Here onwards, we have to call it as a Southern blotting technique. Here we need to call it as a Southern blotting technique, because here we will use one alkali, a buffer, sodium hydroxide we use. This method actually developed by Southern. His full name I am not remembering. I will let you know. The Southern, he only has developed. If you remember, you may share, no teachers, no problem. So then he has run that gel electrophoresis. Then the gel has to be taken. The gel imprint has to be taken into a nitrocellulose membrane. In that membrane, we will be adding one this radio label material. Any radioactive elements we will be adding. Student definitely must write. So far they were calling as a DNA fragment. After adding the radio label, we have to call it as a probe only, because in the exam there is a question, what is a probe? The probe is a radio labeled DNA fragment. Student must be able to answer that. Then probing will be done. After adding the probe, hybridization technique, we need to view it under the X-ray, the auto radiography we can view. When we view that, we will get the different bands. We need to compare the different bands of the sample collected from different people, and we have to see the similarity in that. These 6 steps student must definitely memorize and understand very clear manner. In exam also, mostly based on the 6 steps only questions are asked. Then the PCR, when running also, the PCR also plays a very important role, because whenever we are collecting the DNA, mostly as some very extreme cases, only one single strand of DNA also we may find. That is not enough. If that is goes some project, the experiment goes wrong, we won't have more sample. For the safety point of view, what we do, before itself we make them multiple copies of that particular DNA sample using PCR technique. Also we are doing that, which we already know very well. PCR will, when we are studying biotechnology, at the time we'll be discussing very well. So this is the 6 steps. Teachers kindly, already it is there in the reader. This we need to put it in the mind of children in a very, they need to understand it clearly. Then only they will be able to write all the questions based on this particular topic. The steps, summarized steps like isolation of DNA, digestion using restriction enzyme, then separation of the fragment using gel electrophoresis, then using that on synthetic membrane, we need to transfer that, then we need to hybridize that, then we need to we need to detect the band using auto radiography. This is the entire sequence. What is the application of this particular DNA fingerprinting? Student must understand there are a lot of courses are available for forensic studies. There is a separate university is there for forensic studies. Student may make up the career in that also. So we need to tell them here itself. not only for paternity testing, the forensics. Yes,
Give
ma'am.
me information about northern and western blotting also. We can add one line. I used to add along with the
Yes.
southern blotting.
Yes, ma'am. Yes, ma'am.
Northern and western blotting also I used to add. Actually, southern
You are
blotting.
adding. Yes, yes. That is very good only. Usually
One
we
word
can
we
add.
to the- Yeah.
Yes, yes. That will make the children to understand in a very nice, in an interesting way. It will be there. But a few topics we are not able to add because of the time constraint we have. This chapter we need to complete within 1 month or
Southern
that end.
blotting for DNA extraction, northern blotting for
Yes,
RNA extraction, western blotting for
very
protein.
nice.
So we
Excellent,
can
ma'am. Very nice.
information like there are ways to extract even that also. They should not think that
Very
only
nice.
DNA has to. We can tell that also.
Very good, ma'am. I will I will add it into my class also hereafter. Good. Thank you. So that forensic tools, the paternity, students always think that only for paternity testing, this is used. That student must be explained. Not only for that, even if one person is murdered, if the weapon is there somewhere, if blood stain is there in both the areas, we need to find match that whether that blood stain taken from this weapon and from the person body, whether both are the same only or different. These and all has to be done only by forensic tool. Various, especially in the murder cases, or even if you find a little sample of their hair sample or a blood sample of the culprit, we will be able to find, we will try to find out who is the culprit. So these things and all we need to explain to the children. Not only in that level, even the phylogenetic relationship between different animals, how much we are related to a dog or to a cat or to a monkey, all these things are different apes also we can study using the DNA fingerprinting, because the repetition will be in a very progressive. When we go from lower animals to higher animal, the repetition, that VNTR will be always going in a progressive manner. From that we will be able to do that also. And very importantly, the population, the diversity, the population genetics, we call it as population genetics generally. To study that also, the DNA fingerprinting is very, very important. Why only some group of people will have the Bombay blood group? Why only some of the groups in India will have the blue color eyes? Why only some of the group of people, those who are actually African descendant, they are living in India, they are only participating in the many athletes event? Why? So like that, when we want to know, for that also actually DNA fingerprinting is very, very important. And of course, definitely a very important in the exam last sample paper they were given in the term 1, that what is the paternity testing, how it can be done. There was a question on multiple choice based. So that also can be explained to the children. So that's all about fingerprinting. If you want to discuss anything, you can discuss, teachers. You can say.
Sir, why most of the people here we have ABO blood grouping types, sir?
So our descendant, we are ABO blood group. We don't have Bombay blood group, you are telling, ma'am?
No, the why? What is the reason in our country we have mostly that kind of blood grouping people?
Because
only descendants, or
yes.
if there is any other reason?
Our Indian descendant, our genetic pool is mostly undisturbed. Whatever the gene we are having right from the beginning, it is because of the customs of marrying in the within the same the community or same thing we are following for many ages. Because of that, our DNA is mostly, the genetic pool is remain intact for the maximum. But if you go to that other countries, the genetic pool will actually disturbed very easily. Say for example, if an African marries to an American, the genetic combination, a completely new set of genetic combination will be introduced into the family. So there it is possible that genetic pool is disturbed, and as a result, new genetic variations are introduced. But in India, for a long time, our genetic pool is not disturbed. Whatever the descendant, our African descendant, we are having the ABO blood group still remain intact, not only for ABO blood group, even for the blue color eyes. It is very rare in India. It's also because of that particular reason only. Yes, teachers. So how did you feel about the classes? Is it going somewhat interesting or boring, teachers?
No, sir, interesting
Okay.
only.
It's interesting, sir.
Yes.
We are
Thank you,
gathering
thank you,
more
teacher.
knowledge.
Thank you,
Also, we
ma'am.
are
Thank you
gathering
for
new
that.
information from you, sir.
Thank you for giving that privilege. Even more, I would like to share when time permits. So
Okay,
tomorrow
sir.
is not there, class is not there, and 16th onwards again it is starting. Teachers, ma'am also will be taking classes. Ma'am also will be taking some seminars. So
Okay.
whatever other classes which I am going to take, the biotechnology shall we discuss?
Yes, sir.
Biotechnology.
Molecular
Yeah.
basis of inheritance, sir.
Okay, ma'am. So I will
Because so
biotechnology.
many examples are there in genetics also. Many
Okay, ma'am.
examples are here. I want to.
So
Have
you
an
are
idea.
talking about that
It's
principles of
like
inheritance, ma'am.
molecular basis of inheritance and biotechnology. Now,
Yes.
what you are discussing versus what we are, when we already know, it's very easy for us to understand. But when we introduce certain topics to the children, it is purely new to them.
Yes, ma'am.
So I need some
Definitely.
easiest way to deliver. That's what,
Sure, ma'am.
something to relate it with.
Okay.
Like how when we deal with botany studies and all, we have something to show, something to relate. Like few examples, if you teach, it will be better.
Okay, ma'am. Definitely. Molecular
Like this
basis.
DNA translation. We are,
Okay,
yes,
ma'am.
sir. Biotechnology also, especially example. Related
Biotechnology
examples.
principle and processes.
Yes, sir.
Molecular basis of inheritance.
Vectors, sir.
Over,
Even
sir,
in
now today.
steps also not an issue. Vectors, we are getting doubts with
Okay,
vectors.
okay, ma'am. Vector.
Yes.
Oh, fifth chapter, sir. Now.
Molecular basis of inheritance. Ma'am is covering the transcription. Already ma'am has decided that she will be taking
Okay.
transcription. So other things, whatever is there, I will just discuss with ma'am whatever topic she is going to take. Other things I will be covering that. So anything also, any specifically is there, you can let us know. And there will be a assessment for you all teachers. Sorry to say that. I hope you know already.
At the
At
end
the
of
end
No,
of this,
no, sir.
on the 30th of May, it will be there, I believe so, teachers, on 30th. Not
So
too much.
only
ma'am?
children are having biology exam, sir.
On thought, yes,
On
on 30th we
30th
have our
only.
exam. Yes, we
But
have
we-
separately. Yeah, very only 50 marks, ma'am. Only for 50 marks.
No issues, sir.
There's only is not too much. I thought of having only descriptive type, very simple 2 mark, 3 mark, like that. Very simple we will have.
Okay, sir. Okay, sir.
It won't be so much this. Only what is the cover, like how we can teach them in that way only. There won't be like more content and all, will not, very less only. It will be like how can we teach them? How come we do that? In that way only the questions will be there. Actually, I was asked to inform it to you. That's why just informing now itself. And also the video will be recorded. That will be shared with you after the end of all the 10 sessions are over. That also you can make use of it. Anyway, from 21st onwards, we are all are going to be very busy. We have no time from 21st onwards.
Yes,
Our exam is there. We need to give revision for our children.
Yes, sir. Yes, sir.
Yes, it take time. And on 30th, the question, as I already said, it won't be like depth of the subject or anything. It will be more generalized questions only, the teaching methodology oriented only. So you can
Then
do
what
that.
you will assess and you will tell us the marks, sir. What is the procedure?
What is? Sorry,
You will
ma'am?
assess and you will tell us the marks, sir.
Assessment? Sorry, ma'am, voice is breaking actually.
It will be like written assessment only, no, sir.
Yes, ma'am, written only.
Yes, sir.
In your school, it will be conducted for different subjects, ma'am. And in a
Yes.
paper, pen and paper test only. Then it will be couriered to the HO, it seems. Then it will come to us. So in that way, it will go on as a assessment for all subject or whatever the seminar attended. For all the, it is very common only. So biotechnology, next week onwards, we'll be seeing biotechnology evolution, molecular basis, in which what are all the examples, like today's session, like examples, like how we can put it in the mind of students. That is the expected. Definitely, I will work for that, ma'am. So anything else is there, you can ask. Otherwise, we'll wind off. Amada, ma'am, I never saw you, ma'am. You're from which school, ma'am?
Sir, I am from KRS, sir.
Okay, ma'am. And Saraswati ma'am.
Yes, I too also belong to KRS, sir. Both are handling.
Okay, okay, ma'am. Nice to know.
Sir,
Thank
tell
you.
about yourself, sir.
I am very simple. Very nothing much to say about myself, ma'am. I am Himaninath Sharma.
Simple, sir, but tell about your working experience.
Yes, yes.
Work
Myself, Himaninath Sharma. I am working as a PGT biology in DAV Bell School, Ranipet. Previously, I have been working